Breaking news in the field of neuroscience and artificial neural networks, where Professor Sebastian Sung won the Pradel Award by playing a central role in pushing connectomics from theory to practice, where he championed the use of artificial neural networks for large-scale reconstruction and leading collaborative effort that demonstrated the feasibility of biologically meaningful data at scale. And his work helped transform connectomics from a speculative goal into operational science. And today we're going to cover that topic and a little bit more about those winnings as well as other type of science that are related to this. The modern field of connectomics emerged from a simple but very daunting question. Can understanding the exact wiring of the brain reveal how intelligence, perception, and behavior arise from biological tissues? For much of the 20th century, neuroscience focused on individual neurons or local circuits or average population activity because the brain's full structural complexity was effectively invisible to us, while electrophysiological phenomena and imaging techniques revealed how neurons fire, they provided only indirect clues as to how information flows through the neural networks of the brain itself. The missing piece here was a comprehensive map of connections of synaptic neurons' resolutions. With that map, theories of neural computation, for example, set on assumptions rather than direct evidence per se. Our earlier attempts at mapping the neural circuits within the brain relied primarily on, for example, painstaking manual tracing of neurons in electron microscopy images, for example. Even mapping a tiny fraction of a brain required years and years of labor. The scale problem was extremely overwhelming to us. A single cubic millimeter of cortex of a brain, for example, contains tens of thousands of neurons and hundreds of millions of synapses, and producing petabyte of imaging data is quite daunting. And for decades, this bottleneck limited connectomics to small organisms and partial circuits, as in the brain, for example. The turning point here came with advances in machine learning and computer vision made it possible for us to automate large portions of reconstruction data processes. The key shift here, method-wise, was the application of convolutional neural networks to segment and trace neuronal structure in, for example, electrons data. These models were trained to recognize cell membranes, for example, or synaptic junctions, or axons, or dendrites, for that matter, across thousands of image slices. Rather than treating images as static pictures, the algorithms here of neural networks learn the continuity rules that allow neural processes in the brain to be followed through three-dimensional spaces as it happens in, for example, a human brain. This automation transformed the connectomics from a manual craft into a scalable computational pipeline. Human experts in this case shifted from tracing every structure to validating, correcting, and refining machine-generated reconstructions of the data. As these methods matured, researchers began integrating structural reconstruction with functional measurements, for example. This integration allowed scientists to ask not only which neurons connect, but also how those connections, neural connections, relate to real neural activity during perception or behavior, for example. One of the most significant outcomes of this approach was the reconstructions of a cubic millimeter of mouse visual cortex through the machine intelligence from cortical networks machines in this case. This dataset linked dense structural connectivity with recorded neural responses to visual stimuli. For the first time ever, researchers could then examine how specific wiring patterns correspond to tuning properties, response correlations, and circuit motifs underlying vision, for example. The findings from this work challenge several long-held assumptions in neuroscience. For example, rather than revealing a simple repeating canonical circuit, the data showed remarkable diversity in connectivity even among nearby neurons of similar type. In this case, synaptic strength and connection patterns varied in ways that suggested flexible, experience-dependent organization rather than rigid design within the brain. This variability implied that learning and development shape cortical circuits at a finer structural level than previously anticipated. It also suggested that population-level function emerges from heterogeneous microstructure in the brain rather than uniform modules within the brain structure. Parallel advances occurred in the study of smaller nervous system where full reconstruction became feasible. For example, the Flywire Consortium completed the first synaptic level wiring diagram of the fruit fly brain, encompassing approximately 140,000 neurons and tens of millions of synapses here. This achievement provided a complete structural reference for a behaving animal with rich sensory and motor capabilities. Researchers can now trace information flow across entire sensory pathways, identifying recurring loops involved in decision making, for example, as well as linking genetic cell types to precise circuit roles. The fly connectome has already produced concrete biological insights for us. For example, it has revealed unexpected long-range connections between brain regions previously thought to be functionally separate from one another. It has also clarified how multi-sensory integration occurs through specific convergence patterns rather than diffuse mixing. It has also allowed researchers to test competing theories of learning and memory by examining how synapses are organized within and across circuits of the brains of these flies. And because the fly is genetically accessible, these structural findings can be then paired with targeted manipulations, such as closing the loop between anatomy, function, as well as behavior. At a methodology level, these projects establish a new scientific workflow where large-scale electron microscopy produces massive data sets for us. Deep learning models, for example, perform initial segmentation and tracing, but human expert and distributed contribution validate and correct these errors on the other hand. The results of connectomes are stored in open platform, for example, where it allows interactive exploration or annotation or hypothesis testing from different researchers. This combination of automation is massive, where human oversight and open infrastructure represent a new mode of biological science that resembles large physics collaborations more than traditional lab-based neuroscience, for example. And the ramification of this work extends beyond mapping specific brains. At a theoretical level, connectomics provides constraints trained on models of neural computation, for example, where artificial neural networks are often loosely inspired by biology, but real wiring diagrams reveal connectivity patterns that differ significantly from your standard architectures of the brain. Dense recurrence, for example, or cell type specific targeting and structure motives challenge simplified feed forward assumptions that we've had so far. These insights are beginning to influence biological, grounded machine learning models and neuromorphic engineering as well. Looking forward, efforts are underway to extend these methodologies to entire mammalian brains. Mapping a whole mouse brain, for example, would represent another qualitative leap, enabling system level analysis of distributed cognition, internal states, as well as learning. While technical and computational challenges remain immense, the trajectory established by recent success suggests that such goals are no longer out of reach and just in our dreams. To sum up, the significance of this study of work lies not in single data set, but in a transformation of neuroscience itself by making neural wiring visible, quantifiable, and linkable to function. Connectomics has shifted the field of neuroscience from indirect inference to direct structural explanation, where the long term ramification will shape how intelligence will be studied, modeled or remodeled, and understood across biology as well as the artificial neural network systems alike. It's important to note that the connectomics work emerging from Princeton does not treat artificial intelligence as a replacement for biological intelligence, nor does it suggest computational systems are themselves models of mind simply because they are effective, as we covered in last video. Instead, computation is used in a far more constrained and disciplined role, in that it is used as an instrument for revealing the physical structure in the brain in which cognition is instantiated, where artificial intelligence in this context function as a microscope rather than a mind itself. Its purpose is to make visible what would otherwise remain inaccessible to us, namely the detailed organization of neural circuits within the brain that give rise to perception, memory, behavior, and so forth. This distinction matters because it avoids the common conceptual collapse in modern AI discourse when artificial systems generate fluent language or accurate predictions. They're often taken to be partial realizations of cognition itself. But connectomics resists this temptation. The neural network used to segment electron images, for example, are not proposed as explanatory models of thoughts or a replacement of the human cognition. In fact, their success does not imply cognitive equivalence with the brain. Instead, their value lies in being a tool where it enables the restructuring of real biological network, a tool to human researchers, so to speak, at a scale and a resolution that human effort alone in the past could really never achieve. So computation here serves as a tool for us to access these large-scale data for research to be able to do their job, and not for it to replace the human cognition or the human brain itself. Essentially, using these artificial neural networks for large-scale biological data help us collect these physical records of biological data as to how neurons, for example, are actually connected down to individual synapses. This allows, for example, neuroscientists to ground theories of cognition in the material organization of living systems rather than inferring mechanism from behavior to output alone, as we've done in the lab so far. In this sense, connectomics pushes back against thinner, output driven theories of mind by insisting that structure of the brain is not incidental, but it is fundamental. And seeing this way, connectomics exemplifies what computation looks like when it is used correctly in cognitive sciences, where it does not claim to explain the mind by building artificial analogs that perform well. In fact, it uses computational power to uncover the constraint or regularities or complexities of biological intelligence as it exists in nature. It is very important to note that it's far from contradicting a theoretical critique of AI overreach, where this approach actually supports it. And it shows that computation is most powerful not when it replaces the human cognition, but when it helps us see where cognition truly lives and how it is physically organized within our biological systems. Lastly, from the perspective of our discussions about symbiotic intelligence, large-scale connectomics introduce a crucial conceptual shift. It treats intelligence as something that is not merely expressed through activity, but materially instantiated in structure. By making neural wiring diagrams explicit and computable, connectomics provides a template as to how biological intelligence can be externalized, interfaced with, and eventually extended beyond the brain and the biological entities of us itself. In symbiotic intelligence system, for example, biological signals in real time and machine intelligence in real time do not operate in isolation, but co-evolve through shared representations and constraint. Connectomics maps here, for example, offer one of the first concrete substrate where such shared representations become possible, enabling future systems in which biological organization informs machine intelligence in real time, and machine models, in turn, help interpret and augment biological signals in itself. In this sense, connectomics does not just advance neuroscience, it actually lays foundational groundwork for hybrid intelligence between biological and computational artificial intelligence systems in which structure, signal, and meaning are jointly negotiated between living as well as artificial agent systems. I hope you liked the topic today. Thanks for watching, sharing, subscribing, and liking. As always, I'll add the link to this paper below. I also wanted to mention that Symbiotic Intelligence is now also available on Kindle because some of you asked. Paperback was a little bit, I guess, too heavy for people, but now it's also available on Kindle, and I will add the link below as well. Thanks, and see you next week.